again for our afternoon session. I apologize for the panel starting 15 minutes late. We all went to lunch together, and we were waiting for the check. What can I say? So I guess our next presentation is from Patty on non-targeted chemical analysis. Go ahead, Patty.
So good afternoon. Thank you for continuing with the meeting with us. The next section we're going to go through is the non-target chemical analysis, which is a crucial part for the field characterization study for synthetic turf field and for playground. And in this section, Dr. Randy Magdalena from the Lawrence Berkeley National Lab and I, Patty Wong, will be presenting. So a section of introduction, the field characterization study. Here's the timeline again. We start in 2015, and now we have our fourth meeting in 2019 in May. And we start our sample collection in field and playground. And we, under the guidance from the panel, we have developed our protocol, modified accordingly, and we went in and sampled 35 field and 4 playground. We finished in summer 2018, and we have discussed the data last year. Using the manufacturer sample from multiple recycle facility, scientists from Lawrence Berkeley National Lab developed target and non-target analysis of VOC, and we are currently working on other classes of chemical. The VOC analysis of the field air sample and inorganic analysis of the field crumb rubber sample has been completed. And now we are working on the non-targeted chemical analysis of the crumb rubber extracts. The non-target analysis is used to expand our knowledge on the chemical composition of field samples. The list of chemical targets that we identify in this process will be selected based on priority and then confirmed using reference standard. And the concentration quantified by using the standard can be used to derive the exposure, and also the chemical identity will be used to assess exposure of chemical on field and playgrounds. Okay. The chemical identified in non-targeted analysis will be prioritized. I already say it. I think that's it. So the target chemical will be used to guide our bioaccessibility measurement, as well as for the analysis of our SVOC sample, the particle sample, and the crumb rubber sample, and used to quantify the concentration for potential exposure calculation. So before we go on, I would like to go a little bit about the OEHHA tire-related chemical database, because it's one of the fundamental lists we're using for this non-target analysis. As I mentioned in the overview, OEHHA compiled tire-related chemical lists to assist our targeted and non-targeted chemical analysis of field samples. The list contains chemicals that are reported from literature, reports from government, literature paper from scientists, and on the tire-related study, on turf study. And I want to note that not all the study actually confirm the chemical. Some are chemical suspect, they do through the database match. Some are confirmed. We also communicate with the federal agencies that are working on the crumb rubber study, and we obtain information from them to expand our list. We also have obtained information from the Tire Manufacturer Association and the International Carbon Black Association on ingredients going into tire manufacturing process. we search the internet to look for chemicals that are advertised as used for manufacturing tire. And we put these chemicals into our tire-related chemical database. And as an ongoing process, the result coming from the non-target analysis, the chemical will be entered into this database to expand our knowledge on the tire-related chemical. And it will be used to guide our field sample analysis. So, as I said, both the targeted and non-targeted analysis are ongoing. The goal is to identify chemicals that will be analyzed in all the field sample. And we have conducted targeted chemical analysis in these class of chemical, including polycyclic aromatic hydrocarbons using gas chromatography mass spectrometry, GCMS, in selective ion monitoring mode, the SIM mode. We also conducted non-target analysis on the VOC in air, and we selected the chemical for our target analysis of the air sample obtained from field. We have done the inorganic analysis on the chrome rubber sample from field. So for non-target analysis, the classes of chemical we are looking at is the volatile organic chemicals, semi-volatile organic chemicals, continue with the polycyclic aromatic hydrocarbons. These are the chemical potentially in the emission from chrome rubber as well as from the solvent extract of the chrome rubber. So we are analyzing it based on different setting of the GC that can achieve for different class of chemical. There's also another class of chemical, which is the polar organic chemicals, don't usually behave well in the GCMS analysis. We are using LCMS, liquid chromatography mass spectrometry analysis to look at these polar solvent extract of the chrome rubber. So I would like to have an overview on the workflow for the overall non-target analysis we are processing. Each step will be discussed in depth in the following discussion. So chrome rubber, we obtain it from the manufacturer as well as from the field. We create composite sample from the field. So they contain a chemical of diverse property, from very water-soluble organics, which can be charged. These chemicals usually are less volatile, to the more non-polar, less polar chemical or non-polar organic chemical. They can be of various level of volatility. So VOC, SVOC, pHs, they have various volatility. So we subject the chrome rubber sample to different experimental setup to collect emission of volatile organic chemicals from these sample, and then the vapor was analyzed by using GCMS for non-targeted chemical analysis. And using organic solvent extraction, we analyzed the non-volatile, semi-volatile, and the volatile chemical using GCMS under different setting, also for the non-targeted chemical analysis. The results of these analysis is subject through suspect screening through database. They go through the NIST database search for potential or tentatively identified chemicals, and we compile a list of these chemicals. And detail of these chemicals is in the binder material for today's meeting. We also extract the chrome rubber using polar solvent. Now we are looking at more aqueous with a 10% methanol in water, try to extract out the polar constituent in the rubber. And like I said, this chemical is being analyzed or has been analyzed using LCMS. Unlike the GCMS, which has a very well-established protocol and database for doing non-targeted analysis, LC, the liquid chromatography mass spectrometry for non-target analysis, is kind of in an emerging field, and it requires protocol development to fit our study. And in this section, we'll go through how we analyze these chemicals. And then at the end, we consolidate these chemicals into tentatively identified chemical lists. And by going through prioritization, buying the reference standard, we confirm some of the chemical, and the process is still ongoing. Here is a summary table on the various matrix or sample that we have prepared. We have prepared polar extract, non-polar extract, and also from the non-polar extract, as well as from some of the emission, we look at the pHs. So, and then we have aldehyde ketones, and then VOCs that we look at in the field air. And each of these matrix or chemical class, we use different instrumental analysis to consider, to analyze and come up with the chemical potentially or confirm being in the matrix. So this is the introduction.
When you say confirmed versus unconfirmed, what exactly does that mean in this context? And let me guess and then see if I'm right. Does that mean that from the various spectra and so on, you can identify what the peak is? Is that what confirmed means or not?
We'll go for more detail yet, but there's a little bit difference between confirmed and not unconfirmed. When you first have the LCMS, you go through, you come up with a spectrum with all the peaks. And then you can go for a database to try to identify what is that chemical. And without putting in a real chemical reference standard into the same equipment, is called as a tentatively identified, so it's unconfirmed. There can be potential some chemical with similar pattern because they are analog of each other. They have different, just a little bit difference in structure, but it's not actually the chemical. They can give out similar pattern on the mass spectrometry. So that's what we call those unconfirmed. But once we inject it through the analysis, we match the spectrum, it looks correct on the retention time, which will go in more detail, and the spectral data, then we call it a confirmed chemical.
Okay, thank you.
Sure.
Dr. Bennett.
I had a question. So it looks like you're doing a sample of new tire crumb rubber and then a sample that's a composite to do the non-target identification and then develop a target list. After the target list and the standards have been bought, are you planning on analyzing multiple use samples to look at variability in levels, or are you only ever analyzing the composite?
So the composite, let's roll back a few years ago when we first started meeting. We heard it loudly from it's a very, very good advice that we should look at what is in the rubber before we move on. So we start the solvent extraction. And this is, the manufacturing sample is before it go into the field. And then we create 2 composite samples from 4 different fields each. So it's 8 fields. It's to help us identify the target. And then once we get a target list, we buy the reference, and then we will analyze each individual field, which we sampled last year with different age, different location.
Great.
Try to look for pattern. Yep.
Okay, great. That's what I thought. I just wanted to make sure.
Yep.
Okay, so it's my turn.
Dr. Madalena, yes, go ahead.
Thanks for getting us started. It's good to be able to be back in front of you guys today. We always learn a lot from this end of the table, from your perspective. Today, at lunch, I asked Patty, I suggested that if I was sitting in your seat, the first thing I wanted to see is, so show me the numbers. Let's stop talking methods and show me the numbers. But for this particular study, that comes at the end. And you're going to get the numbers, but that's going to come at the end. And so the point of these presentations, although they seem to be very method-centric, they're really designed to try and make sure we fully vet our approach. When those numbers do come out, then we've built them on a very solid foundation. So that's kind of my intro. I'm going to talk and go into a little more depth about what Patty suggested is our roadmap, our plan. And you'll see increasing levels of sort of complexity as we cycle through this. The VOC analysis was probably a fairly simple analysis to be done, and I'll talk more about that, but it was in a controlled environment before we even went to the field. What we're calling the SVOC analysis, it gets much more complicated very quickly, and so I wanted to kind of build up slowly into the process for non-targeted analysis, the identifying what's there when you don't know what's there, the process we're taking. So I wanted to build up slowly. So I will be switching back and forth periodically between some of the results from the targeted analysis as a comparison, just to make a few points along the way. So here we go. Here's an overview of the presentation. I remember you said talk straight into the mic, so I'll stop looking at you and I'll look at the mic. Sample collection methods, I'm going to go over that. Again, we've done this before, and you've all seen this before, but it's important, I think, to package up what we're going to talk about in the non-targeted analysis with where these samples come from, because ultimately that's what's important. Are the numbers at the end of the day, are they really representative and relevant to the problem at hand? So we did some laboratory-based tests, including controlled chamber emission studies, some direct thermal desorption. The stir bar extraction is like the small step towards our availability studies. So the stir bar extraction was done, and we did some aging as well in that laboratory-based study, and then went to the field and spent a fair amount of time in the field covering a lot of different locations. So the samples collected from the field are both from the surface and from the air. So you have direct crumb material collected from the fields, and you also have air, which would capture volatilization. And I'll talk a little bit about a question this morning of integrating times and spatial variability. So we'll make sure we have a good understanding of that, and then go from there. Now we've got samples in the cabinet. We go to the analysis, and the first step is extraction. And so I'll talk a little bit about those steps before I even come to the data analysis part. And that's where we're going to really focus most of our attention. I'll say it now, and then I'll try and justify it as we move forward. The whole point of this is to create a very strong linkage between the numbers that we find and the chemicals that we identify and the crumb. And so, you know, the environmental samples, it's a little more difficult, but when you actually take the material right out of a manufactured bottle or collect it out of a chamber where all you have in that chamber is the target material, there's a pretty tight linkage. And so that's kind of the whole point, is the sample collection has a lot to do with how relevant the data is. So, what's that? I can see, you guys have it in front of you, right? So, okay, so this is what we tested. Looking at the laboratory-based experiments again, the first thing was the emissions chamber. The only thing that went into that emission chamber was our material of interest. And so we had crumb infill material manufactured fresh from the production lines, and then turf blades with the backing included. So it's a very simple, as far as a complex mixture, but it's a very simple structure. We built these little experimental units in 6-inch squares, the representative depth of a real field, using turf and crumb, and added them in appropriate balance as far as to represent the field, and then put them into our emissions chambers, which are highly controlled systems. They're as far away from the real world as you can get, but we control the temperature, the humidity, the airflow through the system, so we can close the mass balance when we measure chemicals. And what we measure in this system is with high confidence from the material we're testing. And that's an important piece of information as we're moving forward. For the samples collection in the laboratory, the second one we did, and we've talked about this before, again, making the point of the direct connection, we put the crumb material itself into a cell or a small straw, if you will, and put that straw right directly into our instrument. So in this case, it's a one-to-one connection between what's in that straw and what goes into the instrument. So the signal that we get out the other side is directly connected to or linked to that. The third sort of sample collection method, so I'm kind of working through this fairly fast because I think we've all seen this before, but I just wanted to make a quick reminder. The third one was the stir bar extraction, where you have a semi-aqueous phase. It's a mixture of organic and aqueous, but it's on the, we use the word polar and non-polar. Basically, that boils down to whether it likes to be in water or whether it likes to be in oil, and most chemicals sort of fall along a spectrum. So it's really not a very sharp cut line, but for this experiment, we used water with a little bit of methanol in it, and then a stir bar, and in that stir bar sort of acts as a sorbing environment. So as materials extracted from the crumb into the liquid, it's very rapidly taken up into this stir bar, this artificial surface in the water. And that goes directly into, again, one of these sampling tubes or straws that go right into the instrument. Again, a very tight link between the material we're testing and the chromatograms that come out the other end of that test. So then we moved our sampling to the field, and just as a reminder, the question this morning was, what was the integration time? It's always about an hour. So the samples for VOCs were collected for an hour, spread across the day, spread across the field, on field, off field, and at different heights above the field. So we got a lot of spatial, temporal, stratified variability captured in these hour-long samples. The semi-volatile chemicals, because of their nature, they required essentially a 3-hour sample. So at each field, you have some spatial variability because you've got a couple of them spread across the field, you've got one off the field, but it's integrated over a longer period of time because you needed a larger sample volume in order to see these compounds. So in summary, I said it up front, the laboratory sample collection method is so tightly linked to the material of interest that we have a lot of confidence that when we see a peak or when we see a potential chemical, that we know where it came from. The field, it gets more complicated, but in the field we were able to capture things like variability, spatially, temporally, height-wise. So it was important to have those too. And at the end of the day, some of the things that we saw in the chambers, especially for the VOCs, we didn't see them in the field. And there's obviously reasons for that as things decay, as they volatilize, as they transform in the field. You may not see, just because you've identified it in a chamber doesn't mean it follows through the whole process. So at the end of the day, that final list may have things that you've seen before that have fallen off the list because they're not in the field itself. So very quickly, and I don't want to go into a lot of detail here, the sample extraction for the VOCs in this case, all of them were the same, and it was the thermal desorption extraction. So whether it was collected on a stir bar, directly put in the sample tube, or collected air in the field, they all went through the same. So there's no variability in the analysis side of this problem. It's all directly linked. So then we get to the data analysis, and this is where, for the most part, we want the panel to really think hard about whether we're following the right path, particularly for some of these remaining tasks in the SVOCs, the non-target analysis. So as a little background, there are really two approaches. Like we've used the word targeted and non-targeted. Essentially, all that means is if it's a targeted analysis, you're telling me ahead of time what I'm looking for, and then I do my analysis, and then I look at a chromatogram. I'm going to give you sort of the anatomy of a chromatogram on the next slide. I look at that chromatogram and try and find out if that sample or that chemical is in there. For the non-targeted analysis, it's the reverse of that. I have a chromatogram and I have a peak that we think is fairly clean, and we want to try and put a name to that peak. And so you're coming at it from two different directions. Ultimately, the strategies sort of converge on what we mentioned before. You want to have a pure standard at some point that you could run through your system and confirm what you've found. And so with that, I want to just kind of work through sort of a first-year analytical chemistry just to get everybody on the sort of same page on what we can do, and as we, I mean, I guess I could say get down into the weeds or get down into the turf, and how far can we go to really see things before some of the other factors come into play and uncertainties just get too great. At some point, the uncertainties can't be ignored. So when I talk about a chromatogram, this is what I'm talking about, and this is essentially a sample that you put in an instrument, and all of this science is already developed, so in a lot of cases, we're just driving the machine. But there are pieces that or factors that go into this process that are important and will ultimately dictate how much we can figure out from a sample. The first one is the peak resolution. So when you have a chromatogram, you might have one of these peaks that pop up, and it has a really nice, the baseline's running along nice and flat, and then you get a peak and it goes right back to baseline, and you're like, that is a nice peak. And so we know that from a lot of the science that went before that that's a very pure chemical that creates that peak. And so when your detector provides a fingerprint, you can be quite sure it's a very clean fingerprint that would do good in matching. Environmental samples, you rarely get the first, you more often get the second, where you have multiple peaks, whether you know it or not. This example, you can actually see a shoulder on a fairly nice peak. And that shoulder, clearly there are two things there, and there are tools that we have to go in and start separating those, but we don't even always, if the chemicals elute very closely together, this is going to affect our ability to identify them. The second thing that comes into play is what's called signal to noise. And so what you have is in any analysis, you have some baseline variability, and that variability has some scale or some size. So when you have a signal that comes out, or in this case a peak, you can figure out sort of how much your signal is relative to your noise, and you get down below somewhere in the order of 10 to 15, and it gets a lot harder to figure out what's what. So you'll see in some of our samples later on in these talks that there are a lot of peaks in these samples, but in a lot of cases, they're very close to that noise level, and it becomes very difficult without additional chemistry to figure out what those peaks are. The next thing is really an instrument-related limitation, and that's what we call a dynamic range. It's basically how much can you see in a range of concentrations before either the detector gets swamped out or the lower limit of the detection is reached. So it's everything in between there. We can control that a little bit by our sample size, by what we put into our instrument, but we can't always control that across the range. And like in the chromatogram that's on this picture, there are a couple of fairly large peaks, and those peaks are clearly related to chrom. The benzothiazole is the second large peak there, and we've talked about that before. Well, I can't change this analysis I can't bring those small peaks up anymore without saturating the detector from the larger peaks. So again, constraints of working with real samples. The next one that we deal with, and the last one that I'll talk about, is just basically complexity. When I showed you that first peak, how it was nice and well resolved and a good shape, and we have a lot of confidence in that peak being a very pure chemical, and we could feed that into our identification systems. The problem is we're dealing with what we see in this next chromatogram often. We see peaks that aren't well resolved. Not only aren't they well resolved, as you get these baseline humps that come out that are sort of unresolved compounds. And we see a lot of this in the LC analysis, and we've come up with some strategies to go after that. So in summary, the four main things that limit our ability to get down deeper into turf and identify more peaks are the resolution of the chromatogram, and there are limitations on how long you could run your chromatogram, so you don't want to run over like an hour, maybe an hour and some. And you could change columns, but that may get better resolution for some and not the other. So you just optimize and do the best you can. Second one is signal to noise. That's basically worked out. You can't really reduce the noise that much. You could increase the signal. but you're limited by that dynamic range on how much you can increase the signal until you go into chemistry. And chemistry takes a long time and a lot of money, but you can take these samples and effectively split them up and take the bigger things in one path and the smaller things in another. So there are techniques, but they're difficult, time consuming, and sometimes expensive. And then the last one that we just struggle with a lot is the complexity issue. And again, you can handle this to a certain degree with pre-analysis chemistry. You can, the unresolved peak that we often see in these samples are a mixture of alkanes, which are just normal organic chemicals that don't have a lot of functional groups on them. They don't have double bonds. They don't have halogens or anything extra on them, but they have a lot of ways to put themselves together. So you can have a 12-member alkane that goes into all kinds of different shapes depending on how you connect those atoms together. And those all move at just slightly different paces through the column, and you end up with this sort of a blob that is hard to distinguish, and you need to actually mathematically separate that from your analysis. So the point is you can improve and address all of these limitations, but they obviously are going to take some extra time. So now with that background, I'll kind of walk you through the process that we use in our identifying, our non-targeted analysis, identifying compounds. And I'm going to start with the VOCs because that's somewhat of a cleaner system to work in. First example is a fairly dominant peak in this particular analysis, and this is an emissions chamber of a fairly fresh crumb material. And so we've got the peak here that shows up. It's pretty well resolved. It's a good, strong peak. And we pull that out of the instrument and create a mass spectra, or the mass spectrometer creates a mass spectra for us. So when you put this peak into the detector, the molecule always blows up the same. It always breaks apart the same, and there are libraries that you can compare that fingerprint to so you can put an identification on it. So we feed this into the database that we have, and we have one of the more current databases. They revise those periodically, and it gives you information mathematically, which I don't want to go into, on how well it fits with everything in that database and brings to the top the ones that match the best. And so you're actually matching spectrum here. And in this picture, you see the red lines on top, the blue lines on the bottom. Those are the fingerprint. Those are the pieces of that molecule that come out at that given retention time. And going back, the likelihood or the match factor here is really high. It's 940 out of 1,000. So in fact, it's a really, really good match. But again, we're dealing with a very clean spectra. And another thing to look at here is that probability column. That's mathematically the likelihood that it's benzothiazole and not the next best choice. And so you do end up with a really good,
Microphone.
This is Debbie Bennett. What did you do to get from the peak to the fingerprint? I missed that.
Oh, that's internal. That's mathematics. So that peak is being recorded continuously by your software on your instrument. So the instrumental analysis is recording that peak. When that peak goes into the mass spectrometer, which is your detector, it's bombarded with charge. And so this molecule gets excited, and it just can't handle that charge, and it always breaks apart in the same way. And in this case, it breaks apart with this sort of histogram that you see. There's a lot of pieces of it that are 135 units per charge, and so that's how it breaks it apart. And it's this fingerprint of all of these pieces that's recorded in a database by a number of different organizations. We use the NIST's database for this work. And so that information is recorded continuously, and then when you go in to do your non-targeted analysis, then you sort of feed that in either automatically or you say, give me an answer for this particular peak. And that's where it does the matching. This is the results of the matching, and it shows you, again, a very nice, clean peak. The probabilities were really high, so we've got a lot of confidence, but ultimately you'd still want to run a standard, in which case we run a standard to confirm it. So this is kind of what the whole package looks like. All those little windows are pieces of information on how well you did on your non-targeted analysis. That's easy, right? this is, problem is, most environmental samples are not that clean. And so we end up with situations where we have like this little shoulder on the peak on the side here. Again, this is an analysis or a sample that was collected from an emissions chamber from crumb rubber. So it's clearly related to crumb. The weak peaks, they don't resolve very well. The problem with that is they're in the chamber together when you break them apart. And so you've got this mixture of ions that come about. There are a lot of options that we could do to kind of improve this separation, this resolution. I talked about them before, but ultimately the easiest one is just mathematically try and separate those. And we didn't build the software, we just used the software, but it's fairly straightforward as far as it sends all of these ions at any given retention time into a library, and then it statistically matches up traces. So if one mass to charged number goes up and another one goes very closely correlated with it, then it matches that as one compound and throws everything else away, right? So it's just a mathematical approach to cleaning up spectra, and at the same way, the same way we did previously with the raw spectra from the instrument, we can do the same thing with the cleaned up spectra. And that's just a little bit more uncertainty, but it still provides a very nice way to get down to identifying more and more peaks at lower and lower and dirtier levels, or messier and messier chromatograms. So going through the VOC process this way, we ended up back and forth several times. At this stage, there are no more remaining tentative ones on our list. They're all confirmed. And so with the VOCs, and I'll throw into the mix the carbonyls as well. Mostly that's formaldehyde and acetaldehyde, some of the more volatile carbonyls. So I'll throw those in as well because they're also confirmed, and we end up with 78 compounds on our confirmed list now. And I think that list is in the meeting material. So you can refer to that list if you want to get specific names and such. So in summary, I wanted to point out at first that the chemicals that we're measuring just from our the approach of sample collection are highly specific to crumb, particularly in the lab work, but also in the field. They're highly specific to that field, whether it's crumb related or not. The targeted analysis that I talked about, it generally reduces down to chemicals that we have particular interest in or are particularly high in the sample. And we used, at that point, and to address Debbie's question earlier, we basically train the instrument moving forward. Once we have a chemical identified, we don't have to go through this again. It's got a retention time, it's got a fingerprint, it's on our instrument at the very same time every time, and we train the instrument to look at that point and does the work for us. So that's nice. The downside is there's always other things in your analysis. We may do 30 fields, and that 31st field has something else there that we didn't expect. And so you always have to continue to circle back through and look at your chromatograms and make sure there's nothing new popping up that needs your attention. So in the non-targeted analysis, I spoke of the things that kind of control how good we can do with that, and the process of getting from a sample to essentially a name, and then ultimately it is up to us as analytical chemists to go in and confirm that with pure standards if available. That approach on the non-targeted analysis, especially for these more controllable samples, will give you 80 to 90% of the, I'll call it mass, but in reality it's just the response from the device, from your instrument. So it'll cover a good fraction of the response, and the rest of that is so close to the signal to noise threshold that it's really hard to get much better. So that's kind of the overview. Should we stop for questions on the VOCs first? Because okay, so we could stop there and clarification or drill down into questions.
Thank you, Randy. That was very helpful for a non-chemist. I think I understood most of what you said. Any questions or comments right now before we move on? Dr. Bennett.
Yeah.
Sorry.
On the, looking at the VOCs and the tire crumb, you were using 25 degrees C in your chambers. Is there a reason you didn't go higher than that? That seems like a sort of gentle
Yes.
approach.
Yeah, we did go higher than that.
Yeah.
We spoke about that, I think, at the last meeting we had, we circled, sorry, no, that's okay. And we also, the even more difficult, or the more challenging extraction was where we put the crumb directly in the instrument. And
I
in
was
that
gonna
case,
say,
we did go to very high temperature.
Okay, what temperature did you use for those? Once you
That ramped up from representative temperatures in the 40 to 50 C range, all the way up, I believe, to 150 C.
Okay.
You go much farther, you start getting thermal decomposition. and that's
Okay,
really
even
not
at
relevant.
that point, did you get thermal decomposition, or?
It was approaching that. It was definitely approaching that. The things you're starting to see were
Okay,
indicative of what you would think of a breakdown product, and not necessarily just volatilizing off of the system. So it's again, it's a judgment call, but all of those peaks filtered back into our decision process to kind of identify what we wanted to target. And then-
so,
Yeah, go ahead.
okay, so the other question is that you said that, was it in these rubber, crumb rubber samples, were you able to identify 80 to 90% of the peaks? I mean, are all of those peaks actually in libraries that you can identify? Or were there a lot of spectra that it was just like, we don't have that in our database?
In most cases, they are in the database.
Okay.
Surprisingly, they are. You can identify them using the approach I talked about down to the isomer level, which means a chemical that has
Right.
a very similar structure, like the example I showed you with the benzene ring and the 3 methyl groups. It could have been any one of 3 different structures, and you just can't tell them apart.
Yeah,
because you
back in the
move that
olden
around.
days, they didn't
Right.
have so many spectra in
Yeah.
those databases, but now.
It's tremendously rich.
But it's rich, okay.
If you find it in manufacturing or in the environment, there's a good chance it'll be in one
cool.
of these databases.
That's great.
At this stage.
Dr. Bennett?
So am I correct in interpreting on slide 20, you were able to you found roughly 67 peaks and you were able to buy the standard and confirm them all? Or there's some that were unconfirmed and so you crossed them off because they weren't what you thought they were?
No, in this case, it's the first. We did, and in fact,
Well,
we had a lot of them already. We see a lot of things anyways, and we had standards, we had calibrations. A good fraction of those chemicals that we saw, benzothiazole, for example, it's in our normal analysis. I mean, we run it, and so we saw it. So we don't get a lot of credit for doing anything fancy there. That was there. There were a lot more that we identified, and for various reasons, we ended up with the 67-ish peaks that moved forward in the analysis that we
so you ended up buying like half a dozen or dozen additional standards and then confirming?
That's probably a couple of dozen. Additional, yeah,
And then
20
that
to
was pretty
30
much
additional.
everything. Like there weren't any big peaks when you were done that you didn't know what they were.
Correct.
Wow, that's great.
Yeah, and I'm talking now about the controlled samples, the field samples that were a little bit messier, but at the same time, they were still quite low concentration, so that the number of peaks that actually were resolved that we were able to work with.
So
was
the
relatively small.
67 VOCs included when you stuffed the stuff in the tube, when you put it in the chamber,
there, yeah.
and when you did the stir bar.
Right.
From the 3 of those, you had okay.
And we evaluated what we found in the field as well to see if there was anything else standing out that we didn't already have in our list. And for an example, I mean, if there was an alkane or some branched alkane that clearly showed up, there's an approach we use to throw those off the table and not track them further because they're not toxicologically relevant and they don't contribute to any other issues that we're worried about. And so there were some decisions made iteratively back and forth with the various players to wind down to that one list. And in fact, they're all confirmed. And that's kind of the good news. So the next section, you'll see a lot more show up in that column to the right. And so I wanted to start here. So if anybody fell asleep, you would be left with the impression that we did. Yeah, okay, let's move on.
I think Dr. Kyle has
Okay.
a question or comment.
It's related to this. If some other guy did the same thing, would they be able, be able to estimate this 80 to 90% number in the same way? In other words, is there a common way that one, you, a guy like you, no, there's no one like you, but somebody else doing this can quantify what part you aren't able to identify?
That's a great question for the next section. I think we did really good in this section, but as far as the VOCs.
Yeah, well, I'm asking, is there
yes,
a method for that? Because
exactly.
my
That is
point.
not, what I showed you was over-the-counter methods. Definitely, any contract lab could do that.
Including quantifying how much you couldn't find.
They would come up with a similar number, yes.
It would come up similar.
Yeah.
Because I think this is actually part of what needs to be written up as results of this.
Exactly, and that's what, at lunch today, can we, and it's too soon for actually showing numbers. Okay, so we.
We're not going to get to the numbers today. When you said the end, it's not today.
The end of the numbers comes with the report.
Oh, okay.
The numbers
They,
come with the report.
they want to bring us back for another round.
And I'm a contractor, so that's just my understanding of it. I might have misinterpreted it, but my understanding is that the numbers actually, we want to vet this method very well now, and we're looking at chemicals and names. We're working on names of compounds. Those are the important things at this stage, and then the numbers will be in the report itself. And then I think we'll probably be here again one more time, and that'll be the funnest meeting, because that'll have all of the numbers, all the information.
I want to respond back to Randy say he's a contractor.
Thank you.
He's a collaborator and he's the leader of the lab. So he has been the instrumental mastermind on this chemical analysis.
Thank you.
But when the decisions are made, he's a contractor, right?
Yeah.
This is just a quick question. I mean, we had pages of names of chemicals that you gave us, so they have been named. Were there any, so based on what you started out and people said was in the crumb rubber, did it pretty well match what you found? Were there any surprises? Were there any disappointments? Just for those of us who want to hear a little data.
Oh, yeah. Actually, it should not have been a surprise, but the dilution in the real world is such that if you can measure it in a chamber, the chromatogram I demonstrated or illustrated early on was somewhat exciting. I mean, there's a fair amount of stuff there. But when you go to the field and try and track those, it's really just the major 2 or 3 peaks that continue to survive to where you can, where we see, and again, this is qualitative, you see a trend from low to high to where you can really link it to this source. The question about the crumb itself, now that's a little more tricky, and that's what we're going to spend the next couple hours or hour talking about. Yeah, that's a little trickier, and I think there are things that are of interest that we can continue to chase, and I don't want to go into too much detail, but certainly the Europeans.
Chemists always want to continue to trace something.
Right.
or
Give
other.
me a number, and I'll see if I can get below it. The Europeans, I don't know if Patty shared the background, but they just settled on, let's just look at PAHs, because clearly you see PAHs, polycyclic aromatic hydrocarbons, in crumb material, and we do too, right? We see it. And so they just focused on that and took the easy path and said, let's regulate that, and if the crumb is above a number for the PAHs, then we'll track that as our threshold. And so there are different ways from different approaches to deal with that, and that's certainly one we're seeing, and one of the questions I'll ask later is, should we look closer and see more? Because, so anyways, yeah, we'll circle back to that. Did that somewhat answer your question, the curiosity part?
Are we ready to move on to nonpolar constituents? No, it's quite all right, Linda.
Yeah, it's good discussion. Thank you. So the nonpolar chemicals, again, in reality, it's a continuum, right? We use nonpolar to represent those things that we could see on a gas chromatograph, a GC. And so I'll talk a little bit about the approach we use for non-targeted analysis of the chromatograms from the GC of the crumb material. And let's see. So again, I'll follow a similar pattern as I did last time. I want to convince you that the samples we're looking at are relevant, and then I want to kind of sort of fill you in on the extraction approach that we used in order to make sense of the data, because that ultimately will tell you how the numbers we see relate back to the crumb. And then I'll talk it in reverse order this time. I have it listed as targeted and then non-targeted. I'm going to flip that around and just talk about the non-targeted first, and I'll finish with the targeted, because the only targeted at this stage is the polycyclic aromatic hydrocarbons, because it came down a different path. It was thou shalt look for those because those are important. Let's look for those. And that actually, for a chemist, that's always even easier. That's easier to go out at it that way. So sample collection, in this case, we're just looking at the crumb itself. The crumb was either collected from the manufacturer, right out of the bulk products at the manufacturer, and brought to the lab, where it was extracted and analyzed as we received it. There was no sieving or washing or treatment in advance. What we call the installed crumb rubber was harvested from the fields, and we've talked about that at previous meetings as far as how representative the number of fields were geographically and then spatially across the fields, and then moving on and off the field. So we talked a lot about that, but the crumb itself was harvested from the field itself, and again, analyzed as received. So if it had, take that back, if there were big pieces of things in there that were clearly not related to our project, then those things were removed. In general, they didn't make it in there because we were harvesting that material ourselves, but in some cases, there were pieces of paper or other things that just weren't relevant to the study, so those were physically removed. But other than that, everything was analyzed as is. So if it had sand in it, or cork, or other soil material, or pieces of blade, we would analyze it as received. The analysis, extraction and analysis approach followed this pathway that's shown on the screen now, where we started with a known amount of material from the field or from the manufacturer, and we loaded in these little cells that seal up, and they're designed to go to very high temperatures and high pressures. We don't run it at that rigorous of conditions for these particular experiments, but the cells, once they're loaded, are fed into this instrument. And there was the circle at the top left of the picture of the instrument is there to represent the fact that there was some iterative process here, because we didn't want to rely on a lot of cleanup or fractionation or enrichment steps. We wanted it to make this as closely linked from crumb to sample as we could. And so we had the liberty to adjust the amount of material we extracted. So we extracted different amounts of material and ran it through our system until we got that dynamic range sort of fit as much as we could. And then move forward with that volume of material in the cell, the extraction cell. The extraction itself, again, everything went through this one type of an instrument, and it's designed to just, if you've been around for a while, I mean, we used to use a SOX lead extraction, which is a lot of really fancy glassware and boiling. It's like, a distillation type approach. This is somewhat of an automated version of that process. It's faster and cleaner. So we had two different pathways coming out of this instrument. The first one was the organic solvents, what we're calling the non-polar, but it essentially is just what came out in an organic extract. And the second one that Patty will talk about is the semi-aqueous phase extraction, which would go to the liquid chromatography analysis. The instrument we used is listed there. The main point I wanted to show is that, number one, we didn't change the material at all. We didn't do any cleanup or separation or adjustments to the material itself, or fractionation of the extract for that matter. So the extract went right from the instrument right into the analysis. And one important thing about the method that I'm talking about now is the detection limits for this method are on the order of 3 orders of magnitude lower than the detection limit for the volatile organic chemicals. And there's a lot of reasons for that. I don't need to go into too much detail, but you can actually see more in these samples than you can in the volatile organic chemicals for a number of reasons. So the non-targeted analysis, so basically the process we follow for non-targeted analysis is very similar to what I just went through and described to you. The differences are that the molecules are often more complicated because they're bigger, they have more functional groups on them. at much lower concentrations because they're not volatile, or in this case, they're being extracted from something, so we have control over that. But it allows it, but in order to get really good detection limits, you really almost have to look at or look for specific compounds. So we really want to do our best to take things from this targeted from the non-targeted to the targeted column. Because once we get there, then we can drill down really deep and actually get good confident numbers. So that's one of the driving factors to push us down this path, to try and identify as much as we can. But the real challenge in this SVOC, whether it's on the LC side or the GC side, is the complexity of the chromatograms. And the three-dimensional plot sort of shows you all that's going on in this GC mass spec analysis with retention time running along one axis, the height and color of the columns, and you'll see these three-dimensional plots again on another axis, and then the mass to charge ratio going in the last direction there. So you've got all these things happening at once that make it difficult to analyze and identify things. And you really deal with this complexity thing as well, and the complexity changes with whether it's from the manufacturer or whether it's been in the field for a while. Yeah?
Is this like a time of flight instrument or no?
No, that's just software that actually shows you the 3 dimensions. So you're right, you could do this. In fact, what we're going to talk about with the LCMS is a 2-dimensional mass spec, where the first dimension is the non-ionizer or non-fragmented version of the molecule. And then it goes into a second dimension, similar to time of flight, but this instrument's even more sensitive than the time of flight. I don't think that instrument's a time of flight, is it?
It's
It's an ion chamber. Or
it's an ion trap. I don't want to say it wrong. What's the name? Orbit trap. Linear ion.
Yeah, so the instrument, we're not using a time of flight, but I understand your question, and this picture is just the complexity of a standard chromatogram with fragmentation. So you have fragmentation going in the direction. I had to make it smaller so you don't see the axes. I'm sorry about that. But for example, just past the 12 there on the retention time, you see a line of peaks going into the screen. That's a mixture and fragmentation taking place all at the same time. So that makes sense? Okay. So circling back, in the case of the SVOCs, often the extracts are tremendously complex. And in an analytical chemistry lab, you don't always see this complexity because there are often steps taken before the instrument is run. So there are some chemistry steps where you separate things out, you remove things you're not interested in, and then just look for specifics. But for the discovery phase of this and the targeted and non-targeted, or for the non-targeted in particular, we did not want to take anything out ahead of time. And so you get very complicated chromatograms. The field chromatograms tend to be more complicated than the ones from the manufacturer for obvious reasons. You have things settling on the field, you have aging, you have sunlight, things taking place in these samples. So it's creates a lot more variability in what's in the sample, and so you get these unresolved peaks. And as shown on that mass spectra or the 3D spectra previously, they are just complicated, and you have to just work through that complicated mix. Sometimes you get lucky. You still get fairly cleanly resolved peaks, as shown in here, that you can send through the process in a similar way. But when I put this up here, my first glance was, yeah, that's a great peak, but in fact, there's something right there where the circle is as well. And so, again, mathematically, you could send that through the system, and this is commercially available software. In fact, this one is I believe it doesn't even cost if you buy the other database software. This is developed as well. But what this software does, as I talked about with the VOC analysis, I didn't do this with the VOC. So this is a new piece. What this software does, anyways, I think I did talk about it. It deconvolutes the spectra, right? And so it finds things that elute together and rebuilds a spectra, even in a very messy system, and allows you to send that spectra through a library and match the cleaned up spectra. Often and almost always, it's not as good as a clean VOC sample, but it still gets you close. So I'm not going to go through that whole process again, but that's the main tool we use. The next tool that's available is chemistry, and we're trying to, at least in this stage of it, we're trying to avoid that to a certain degree. So I'm going to talk a little bit about the targeted analysis, and then we can go into questions. In the targeted analysis, the reason we can do so good, our labs can do so good, is, and knowing what you're looking for, a good example is the PAHs, the polycyclic aromatic hydrocarbons. Method's been around for a long time. There's an isotopically labeled standard for a large number of these compounds. So you actually put an internal standard in that's closely linked to the chemical that you're looking for, so your targeted compound. In this case, we ran that liquid injection without cleanup and looked for 18 or 19 specific polycyclic aromatic hydrocarbons. There's hundreds of them, right? There's hundreds of them. These 18 or 19 have been used by the EPA for 20 years. They're the poster child for PAHs, and that's what we continue to go back to. But the point is there's a lot more. Whether there's tox data to go with those is another question, but there are a lot more, and this is just an indication of the PAHs in the system. Patty mentioned SIM analysis earlier. And so when we're doing these SVOC analyses and we have a target that we're after, we can train the instrument to just look for that target and ignore everything else. And so you end up with a chromatogram that actually looks fairly clean because you're only looking for a dozen mass ions and ignoring everything else. And so you can really do a nice job getting down to super low levels and femtogram levels, high femtogram, low picogram levels on the column, which is, even when I was in school working for Tom, that was absurd. You couldn't. And so this is good stuff, and it really allows you to go well below what you would need for a risk assessment with high confidence.
Could I ask you just one question so I don't forget it later? You had mentioned the Europeans are focusing on pHs.
Yes.
Are they focusing on a battery or of like 18 like this, or are they focusing on the many hundreds of pHs, or do you know?
They lean towards a smaller set, yeah.
Yeah,
Yeah, they will look, either I don't remember correct, I remember correctly.
ballpark, yeah.
We have a meeting with them. I remember probably it's around 8 chemical,
Okay.
pHs they were looking at. And they actually say now they have to go back and revisit the issue.
Thanks.
Okay, so continuing the targeted analysis, this particular analysis, another point to sort of take home in your decision or in your critique of the methods is that in this particular analysis, because we're just looking for this class of compounds, we miss everything else. And so if I wanted to do phthalates, for example, I would have to do another run, or I would do another run and focus on phthalates, and then another run. And so the instrument time is greatly increased if you're targeting specific classes of compounds. The bonuses or the good side of that is that once you get a targeted analysis, a lot of this is automated. And so that helps kind of balance out the more instrument time, sometimes lead to less analyst time, if that makes sense. So at the end of the day, with the GCMS side, what we're calling the nonpolar extracts and the targeted compounds that we started with, it bumps us up to 130 confirmed compounds. There's a handful in there that are not confirmed, quite a nice handful, 182 peaks that we've gotten tentative identification through the process that I described here that have not been confirmed, and we'll talk in a few minutes on how to prioritize how much we confirm and how much we don't. So, for example, if one of those is an alkane, or I noticed on the table earlier that argon somehow got through that list of unconfirmed compounds, and I don't know how argon got through the list. It came out of the instrument, it was seen by the instrument, and unfortunately I didn't catch it in time. So yeah, there are 182 there. I would say 181 if you remove argon, because I could probably do that right now. But the question is, how do you prioritize quantifying those or targeting those to put them into the other column, and if you do or not. So in summary, targeting chemicals sort of helps us get better vision. It helps us look closer, look at lower concentrations, and with a lot more confidence if it's a targeted compound. The trade-off is when you're targeting things, sometimes you don't see other things. You get under the street lamp that cartoon earlier sort of shows. The sample enrichment is an option for bringing low-level concentrations up, but we were hoping not to need that, and we've been successful thus far not having to. Enrich is a simple way of saying concentrate things. So I evaporate the solvent off and try not to lose the chemicals I'm interested in, and that gives me a higher response. But we're essentially running these compounds as extracted without a lot of those steps. The other one that's fairly easy to implement, but it takes some time, is our concern about that unresolved hump. That would probably be greatly diminished if we did just one cleanup step of the sample, but we would essentially lose all of the alkanes in that one cleanup step. So in this stage of the search and discovery phase, the non-targeted analysis, we chose not to do any cleanup. And the picture you see on the side, unfortunately the scales went away, but for the full scan, you kind of need big peaks to see in the full scan. The selected ion monitoring does some cleanup for you and allows you to just look at specific molecules. So that's kind of the overview of where we're at with our process of populating that table with confirmed and tentative chemicals. And then you've got a long list of chemicals in a table that we're still working back and forth on.
Dr. Avil, Mr. Avil, why don't you go
So I
first?
have a question in terms of your prioritization and going forward with identifying these thus far unidentified peaks, but peaks that you're potentially targeting for more discrete analyses. Are you thinking about looking at these in terms of toxicological classes or based on some biological input, or are you looking at this from a sort of a chemist standpoint that here's a peak that I can't discretely identify, so I want to identify this just to know what it is?
Yeah, I was just handed a note. I'm not sure what it says, but I'll answer anyways, and then I hope I answer it correctly. So when you say a peak, in general, we've on our tentative list, we've got names associated with those peaks, and I think your question was, how do we prioritize confirming those names? And in fact, the toxicology plays into it, the size of the peak plays into that decision. So there's several things that play into that decision. You can scan through the list fairly quickly and find a lot of things that are almost certainly of insignificant toxicological importance, and those could go to a lower priority. There are some things that we just don't know, and there may not be toxicological information for it. So those are sort of in this middle range, and I think we'll look at we'll revisit this topic at the end of the next presentation too, so we can kind of get more information. But that's, my answer is that, yeah, we were trying to prioritize it based on several factors, toxicology being one. So does that answer your question?
I just might jump in, Ed. In the next presentation at the end, slide 16, are the questions for discussion, where they actually want our input on all these questions. So you
that's it.
jumped the gun a bit.
Yeah, so that's what was on the list. I just put the glasses on.
Any other comments or questions? I had a quick clarifying question. So the 32 on the previous slide, those were your targets that you had purchased before you started this whole project, right? And then 182, you haven't purchased any of what you suspect you saw, or the 32 ones that you've already said, okay, we saw this and we want to go back and confirm it?
The 32 are ones that we either had standards in the lab that we were able to confirm it right away, or we purchased those standards because they were, for whatever reason, rose to the top right away.
Okay, so that you've already started prioritizing. You didn't have a target list going in.
No, entirely, not entirely. We didn't have a target, especially for the SVOC side. We didn't have a target list going in because we didn't do SVOCs in the chamber as a sort of a reminder. So we did, going into this, it was a matter of running that extraction and then starting
And then
from
did you not, so the 32 that you confirmed, were those all successful, or did you have some that you're like, ooh, we thought it was this and it wasn't, and it became an unknown at that point? Or were the first 32 all correct?
The 32 are ones that have been correct. They weren't all correct right offhand. There were some that we ran and didn't see. And we'll
Okay.
talk about in the LC side, it actually happened a little more often, where we purchased standards, ran the standards, and thinking we were going to find peak A, but all of a sudden, hey, wait, that's
wasn't.
peak B. It's there. We got lucky. We weren't buying the standard for peak B, which was a smaller peak, but it seemed to match up. So there is some intuition that goes into it as well when you start looking at different versions of benzothiazole, for example, different functional groups on that particular structure. They may be little smaller concentrations, but it's like, hey, that might be there.
And
It
then
may
do
not
you-
be the peak you're looking for, but it could turn out.
So I know with time of flight, if it's got something that's in kind of the halogen column, then it's like your probability of being right is much higher. Is there something similar with the technique that you're using? And then of these 182, do you have a lot of things that have something from the halogen column or no?
I don't think so. Not a lot of halogens.
Yeah, okay.
But a
I mean, I
lot
wouldn't
of,
think so for the chrome rubber.
nitrogen,
So it's going to be all
sulfur.
these weird ones then.
Nitrogen and sulfur, oxygen, obviously, those guys show up.
understand.
And so you got, but not a lot of halogens, no.
Okay.
So maybe just one last question from Dr. Sheldon.
Well, I've got two questions.
Well, two questions.
Okay. So the first one is when you did your extractions, you did an extraction in hexane acetone, and then you did a methanol water. Were those sequential in the same sample? Okay, good, because I was going to say, that's good. Then, when it comes to
No, I shook my head for the radio. No.
What?
Sorry about that. I'm sorry, I realized I shook my head and I was trying to record it. and that didn't help.
Okay. Well, the answer is no, so
Yes.
that you're not, okay, good. Okay, so the next thing is that, on this hump thing, as you say, it's all alkanes. Actually, your last bullet says it all, it makes the performance of the column worse, it degrades the instrument more. I would think that if you fractionated and got rid of all the alkanes, I don't think there are any really toxic alkanes that we're concerned about in that area, that you're going to just make your life easier. And you already have the data that shows, even if you don't throw them out, you're not going to detect anything there. So I would think just getting rid of that alkane hump would give you the opportunity, if there are other compounds there, to identify them. And I think the trade-off between cleaning up and not having to re-clean up and re-clean up and re-clean up your instrument would be a whole lot better. So if you had a question about that later, you have my answer on that. You should just get rid of the alkanes.
Outstanding, thank you. Appreciate that.
And can I ask one more question? What was the percentage of things that you identified versus the total hump of stuff you had there? I bet you it was probably about 5%.
Well, it depends on how you draw the baseline on that hump, right?
Oh.
Because if you
Okay.
draw the baseline and follow the hump and ignore
and ignore the hump?
ignore it a little bit to a certain degree, yeah, it would have to be a very small number if you actually
Okay.
included that in there.
Okay, thanks.
Okay, I think we better move on. Is Patty presenting the next?
So
No break at 1:50.
we are not going to- So I can start talking. We just did a beautiful presentation on how automatic you can do with the GCMS non-target analysis. I'm not saying it's easy. The next picture we're going to show is the non-target analysis of polar constituent. As I said, polar chemical, they behave differently, and they require the liquid chromatography, mass spectrometry, which is not as established for doing non-target analysis. So like I said, most of the polar chemical, because of its high solubility in water, they are not suitable for GCMS, gas chromatography, mass spectrometry analysis. So we choose using the LCMS, the liquid chromatography mass spectrometry. The idea is the LCMS result will complement with the GCMS to look for different portion or different class of chemical, so to provide a comprehensive analysis of the field samples. So, like I said, this is more like a research when we go to the LCMS non-target. To make it more efficient and more standardized, we developed a two-tier non-target approach to analyze the LCMS data. And we also applied advanced computational tools try to improve the success of identifying candidate of unknowns. LC is different because we don't have a data rich on experimental spectrum like the GC. We do have some available, but we'll go into that a little bit later. So with the tier 1, we start with a suspect screening analysis. So we use 3 different databases to look for chemical that basically these 3 databases have a different focus on chemical of interest. The first one is the OEHHA chemical list that we introduced earlier. These are the tire-related chemical. So we are really looking at under the lamppost, whatever people has been reported, and with the current information that we are accumulating. And then the next one we look at is a proprietary software, Compound Discoverer. It itself searched the ChemSpider database, which contains a huge number of chemical structure. And some of these, and many of these are pharmaceutical, pesticides, chemical as well. And we also searched the US EPA database, the chemical dashboard, which searched the DSS Tox database. Is a collection of chemical that's of US EPA's interest. Chemical like pesticide, environmental contaminants, pharmaceutical, food additive, high production volume chemical, etc. So here, I want to illustrate how we do the tier 1 analysis.
It'd help if you could advance the slide. Oh, here, go.
Yeah, I'm going through. For example, this particular chemical we look for is the 1,3-dichlorohexyurea as example. Here's the workflow, how we look for tentatively identify these unknown in the crumb rubber extract. Remember, this is a polar extract. We inject it into the LCMS. The system we choose to use is a linear ion trap, orbit trap system, which provide a very high accuracy on the molecular weight, or molecular mass of the molecular ion. Sorry, I have to take it back. And some of the study has reported this equipment has a very high sensitivity with a detection limit at the level of 0.5 to 20 ppt level of chemical in wastewater. So we inject it to LC, and using the software come with the LCMS Excalibur, we extract the signal and we plot in a 3D. We'll have other 3D chromatogram later to go through more detail. But here's an example. We have a peak at around 30-something minutes, and the mass ion M/Z ratio is 225.1967. So the machine is very accurate. So we take this mass ion, and then we put it through Compound Discoverer. It will convert it back to a neutral mass, and we will search for the ChemSpider. What are the chemical in the database that has this mass ion or this molecular weight? Mm-hmm. We also truncate it to 2 decimal places and go through a molecular mass match in our database. Now we are looking at tie-related chemical. So luckily for this particular chemical, we have 1 hit. It's the dicyclohexyurea. I want to make it.
Oh, yes.
Dicyclohexyurea. And we put this chemical in a tentatively identified chemical list because we haven't confirmed it. To make it enriched, we might not be a Thai-related chemical. We also put it in the US EPA DSS tox database. And now, because of the high accuracy of the molecular ion, we put it through all the decimal place, and we go through the search, and of course, it come up with more than one chemical. So we select which dicyclohexyurea to buy, and we purchase the reference standard, and we have confirmed it based on the retention time as well as the mass spec fingerprint. So for the manufacturer sample, one sample in a single scan in our liquid chromatography mass spec, and of course, it comes up with multiple peaks. And by searching the ChemSpider database using Compound Discoverer, it's more like a semi-automatic search. The database itself has 72 million chemical structure. It comes up with 700,000 possible based on the 225, 224 molecular weights. And we research it through the DSS database, which has 850,000 chemical structure in the database. It comes up with more than 80,000 chemical for that particular molecular weight. And we search through the OEHHA, sorry, I take it back. It was a whole scan, not just a 225. It's the whole scan with all the peaks. It come up with 800,000 possibility of chemicals. You research through the OEHHA Thai-related chemical list. We use Excel to just match all the molecular weights come out, and we find 250 chemical as a possible candidate. And we have select 27 of the chemical for our first attempt to buy and to confirm the unknown chemical. And this standard go through 3 different processes of possible how to analyze it and confirm. I go back to the GCMS to look for if that chemical is there. It go through the LCMS with 2 different ionization methods, the positive versus negative. Some of the chemical structure are more easier to be ionized under the positive mode, versus the other is more in the negative mode. So it depends on the structure of the chemical and the functional group on the chemical. So as we have shown here, this is like, I would say almost impossible, pretty much impossible to go through the 700 or the 80,000 possible chemical to look for what we don't know or what we might know. So because of that, we developed a tier 2 analysis.
Before you go on to tier 2, did you say how you picked those 27 to purchase initially?
Yes, I didn't. That's actually the question at the end, but we did have a lot of discussion back and forth with the lab. Multiple things consider is if the chemical is Thai-related, is there for a reason. Do we see in the manufacturer sample as well as the field sample? Do we see it with a reasonable size peak? And then that's another one. Oh, is this supposed to show up in the LC? Because some of the chemicals shouldn't, it's just not so non-polar. It should not be in the extract of being a polar extract. So with all those factored in, we pull up our first attempt is to let's get some standard and see whether the method works. Yeah.
Thank you. That was very helpful.
I ask a question. I'm not clear on all of this. So here, the number of possible chemicals came when you had truncated the mass of that particular spectra to 2 decimal points. Is that right? So
I said it by mistake. This is a full scan.
it was a full scan,
sample.
but how high resolution was it? Because the higher your resolution, the lower the number of chemicals are going to be. So how high a resolution was your scan?
It's a very high resolution that the scientists who run the instrument tell us
But
1 to 2 ppm difference in accurate mass.
But you said on the slide before that you truncated.
We truncate to match with the OEHHA list. We did not truncate it when we go through the
Oh,
DSS
okay, okay,
search
that's
or
what
the
I was trying
ChemSpider
to.
search.
And then the other thing is that this was in the positive or the negative ion mode?
This is the positive ionization.
So, in fact, you were getting mass fragments, not just the molecular weight the molecular mass. Because a negative ion, you don't break up into mass fragments, is that right?
Though both of them break up for the
But negative
fragmentation as well.
ion is much less susceptible to that. So you are getting all of these things, you're getting many different fragments. So that's also sort of complicating what you're doing. Okay, thank
Yeah,
you.
we
I just
will
wanted to
show-
understand what you were doing. Thanks.
So this is the positive scan of one sample with the full scan with all the peaks, the possibility come out.
Thank you, Patty. Why don't you keep going?
So of the 27 target we put in, so far we have confirmed 18 of them. You can see there's several different classes of chemical, and this is only the positive mode. We have not finished with the negative mode. And we see that there's a benzothiazole group of chemicals. We have acid, we have aldehyde, we have a lot of amine and amide in it. We're not just seeing one specific class of chemical. So, as I said, we need to have a tier 2 to look further down to what we can identify. So in the tier 2, we use cheminformatic tools to assist in identifying candidates. We use both the MS1, which is the molecular mass, the first level of the mass spectrum data. We also use the fragmentation data, which is the MS2 data. Now we're looking at the different fragment of the chemical. And by using the LC analysis, we inject it and visually inspect the peak. We take out the MS2 data. Now we put it into the cheminformatic tool and then look for a spectral match. And then the tool tell us these are the potential candidates and how good they match from 1 to 2 all the way down. And then we pick the top 2 candidates into our tentative identified chemical list and prioritize it and go through the confirmation process. So we have checked out 5 different cheminformatic algorithms. First one is the MathFrag. US EPA is incorporating this particular tool into the DSS tox database. We test it out, and we also want to test out different tools because they have different focus, they have different spectral information. The GNPS, the next one, Global Natural Product Social Molecular Network, focused on natural product. The HCM focused on pharmaceuticals. Compound Discovery has their own little database. And then also the competitive fragmentation model ID, which is a tool that trained on 11 very diverse database. And it collects spectral data from it and also generates in silico spectral data for chemical. So it has a hybrid. So we choose to use the CFMID for the reason here. Here's the results. Since we already have 18 chemical, we have the spectral data, so we know what's that chemical. Now we take those data back into the cheminformatic tool. We have this fragmentation fingerprint. Do you give me the chemical name as the top candidate? So it's a validation method to see how well this model behaves in our chemistry world. Different chemical might have a better database, different database. So we try to test the CFMID. Luckily, CFMID predict pretty well. Of the 18 chemical, 11 of the standards show up as the top candidate. 5 show up as 2, but we do have 5, so 2 show up as the number 2, and 5 is not on the candidate list. Because they have a first screen is what is this molecular weight, and they pull out all the chemical from the database, and then they match it. So the 5 chemical we have are not even in their candidate list. While we use MathFrag, which is a total in silico, they predict the spectrum of data, we have none hit for our 18 chemicals. So that's why we choose the CFMID for tier 2. So I'm going to go through quickly on showing how the data look like, the data we use for our non-targeted analysis, and use the CFMID as our tool to identify these unknown. Okay, so I'm going to let it start spinning. Look at the bottom is the LC chromatogram with a retention time. The predominant peak is at the 27-minute. The y-axis is the intensity of the current. And we thought that we have very sharp peak at 27 minutes. If you look at the 3D chromatogram, they actually consist of at least 5 predominant relative high peak with different molecular M/Z ratio. So that's the beauty of this equipment, has a very high resolution, and even in the LC side don't have the resolution. Being 2 mass spec in tandem, we actually have the MS2 data, and we can have the resolution based on the M/Z ratio. So again, this is the chromatogram for the LC for the manufacturer sample. I want to show you how the field composite sample look like. Again, we see a hump here, just very similar to the GC side. We have a lot of chemical in there. Now the question is, how do we draw the baseline to find out how high the peak is? So we put it in the 3D. We're going to spin it again and look at it. Actually, the hump may not be as bad as the GC issue because we can resolve it by the M/Z ratio. And you can see that within the hump, we have some peak that's pretty sharp, pretty well defined, because we do have the MS2 data in it. And also the M/Z ratio able to resolve the peak a little bit better. And one thing I want to point out here is you see a very different pattern on peaks between the field sample and the manufacturing sample. The predominant peak is different. It's shift to more earlier time. And we have a series of peaks in behind, which we're actually able to see and identify what are those chemicals. So I mentioned about negative mode. We also do negative run. We are in the process of finishing the data. We also collect the MS2 data, the fragmentation data for the negative ion, but we are in the process of analyzing it. Again, you see the LC chromatogram, how it resolves in the column just by retention time. And this is the positive mode for manufacturing sample. We have 2 different composite samples. They look fairly similar in this. And then if you look at it, this is a negative ion mode. There's a shift on a lot of peak. The predominant peak is the later time is different, in a different position. It suggests that because we use the same LC system, if the chemical are there, if it's shown in 2 different ionization modes, it would be still shown by the same retention time. So if they show up on different retention time, we're sure that they're different chemicals. So the data here, the pattern here suggests that we probably should run our field sample in both modes because we are actually capturing different class or different chemicals. So again, let's look at the positive and negative mode for COM2. This is composite sample 2. You can see on the screen, when I look at it, the green side. The green is the positive mode, which is on the screen on the left-hand side now, and the brown, which is the negative mode. And if you look at the 3D, for this particular composite sample, we capture much wider spread of chemical in the negative mode, and also we got a lot more peaked in the negative mode than the positive mode. So this is just to show how complex the LC and also how the technology can help us in identifying the unknown. And so far, we haven't analyzed any tentative chemical for the negative ionization mode. So based on the positive mode, so far we have identified 47 peaks with 47 tentative chemicals in our database. And that brings up to 228 unique chemicals in our tentative unidentified chemical.
So rather than read all these questions, I'll have my colleagues look at this. Do you want to say anything in general about the questions for discussion before we launch into discussion?
Yeah, we are looking at your input on how we approach it, because we try to use the database to screen it, which is a quicker way. But then chemical informatics has their own shortfall, because the model depends on the model focus. You might or might not get the chemical. So we would like to have your input on how we better approach it, or anything that we might have overlooked. Also, we prioritize the chemical based on different factors, and we would like to have your input. Do we miss anything? What is the best way, how we select the chemical in the different level of looking at these tentative chemical? I think that's the main point.
Thank you, Patty and Randy. And I just want to say, for me, I think you did a really great job of going through complex material. And because we have to have time for a public discussion, I'd like to keep our discussion right now to about 10 minutes, and then we'll come back after the public discussion to have I'm sure we won't be done in 10 minutes with these questions, but I think that's a way of making sure that we incorporate the public, and there are probably a number of people both in the audience and on the internet that have been waiting patiently. So would that be okay? So who wants to start off?
I do, because I have a simple one. May I?
You may.
This is all very impressive, and I think I got about 80%, so that was excellent. But there's something that's important about this that we haven't talked about, I think, and that is, in the context of this project, ultimately we have to decide what a level of due diligence is here, for the state to do this. And, it can't be infinite, and it has to be enough. And it has to be the right amount of enough here, because the state's paying to get this done. They're paying people to put this stuff in these things. And so some vocabulary from what you all are doing about how to talk about that, I think it's going to be important. Do you understand what I'm saying? I mean, I'm just going to say that because I don't have a suggestion. I have to mull this a little bit more, but it's an additional thing to just getting these results. It's like, how do we describe how much we did of what the uncertainty is? Thank you.
Did you want to respond to that at all at this point? Okay. All right. Linda.
So there are two things. With the polar organics, it's sort of interesting because, now correct me if I'm wrong, but from my old brain, polars tend to degrade a lot more rapidly than those things that are not in polar. And so my question is that knowing what you do about, the samples look completely different from the crumb and the other, and they look more similar in the others. And so I guess, is there a way you can start to sort of look and say, are all of these tire crumb things really just degrading in the environment to something else? And if they are, then, how do we deal with that? Because that may be something important to consider when and if you actually do the nonpolar analysis. The other thing is when it comes to prioritizing chemicals for confirmation, again, we are trying to look at risk assessment, and a lot of these chemicals don't have the toxicity criteria. And Lauren, you may not agree with this, but I do know that, in EPA, they do structure activity analysis. And, maybe what you want to do with that whole slew of nonpolars and things that you're not dealing with is a structure activity analysis and say which ones are potentially toxic, and then go from there. And so that might be another thought. But both, do you do them because they, do you even bother because they degrade? Or given the ones that have degraded, are any of them potentially toxic? And, then proceed from there.
That's one of the reasons when we sample fields of different ages, and we want to capture if chemical degrade and also environmental deposition into this rubber. And we believe some of the components are deposition, some are degradation. We totally agree. We're actually surprised to see these field sample has so many polar chemical, and they seems to be hanging there, or they are depositing onto it. So, like I said, yeah, like you suggest, it's very good idea to look at the chemical as a class, especially for chemical that has weakened database of toxicity. We are looking into how first of all, we have to find what are these chemicals, and then we're looking how to bin this chemical into different classes, and looking for alternative method on how can we address the toxicity issue here. So everything are going.
Well, and those fields have been in the environment for 1 to 3 to 10 years, and they are sinks for everything that's out in the environment. So, you also have that, which is an issue. And I don't know if you deal with it saying, well, they're a big sink and we have to deal with it, or, this is not really the tire crumb that we're worried about.
I think we're going to need to have a very in-depth discussion on what it means. But definitely, we are looking at it from the exposure point of view. People are exposing to this chemical. That's how, at this point, how we look at it.
Dr. Bennett?
On Linda's comment about the degradation, I think there's some Swiss tools that do degradation products that you can then integrate in with some of these methods and try and look for degradation products. And so maybe taking like your tire related things, running them through the Swiss software that predicts the degradation products, and then looking for those might be a way to help understand what's in the field. So I was just surprised because on the LC, I know that at UC Davis, I work with Dr. Young, who's got one of the LC time of flights. And I know he uses a bunch of Agilent databases for suspect screening on the LC, because he's got like this Agilent Mass Hunter, a forensic toxicology database with 8,000 chemicals, a water contamination one with 1,400. Is this just a different machine and Agilent doesn't have those databases, or is that something that he purchased in addition, or?
This is a thermoscientific
Oh, it's not
so it's a much sensitive than the TOF, and also much more accurate. And the reason we do these two, because LC world doesn't have standardized protocol. Each fragmentation or retention time is going to be method dependent and also your equipment dependent. These databases in general, they collect experimental database, and you can compare the best you can. You might not get 100% match, not like the GC world is so pretty. That's why it's critical that once we get the suspect, we confirm it with our reference standard, then we know exactly this is it. We don't get 100% match on the suspect, but we do get 100% match on our standard versus our sample we suspect. So it's only if you run it through the same equipment. And the CFMID itself do have more than 200,000 unique chemical and has experimental data embedded in it together with spectral data. But they don't necessarily mean this exact same condition. We are running this profile on the solvent, the energy on the equipment. So it's going to generate different pattern.
Okay. And then on the tox data, I'm assuming because you said that you were doing some of the EPA databases, so I'm assuming you're doing all that sort of in vitro ToxCast. You're using that to kind of rank some of the toxicity of these chemicals, because you can kind of pull those databases of those compounds that they've done the high throughput in vitro screening, and that might be a quick way to get some tox prioritization on some of these.
Yeah, we are looking into that. We are collecting those information.
Okay, and then I had a list. I had a comment too on the QSAR tox models, like Linda, and I had a list of ones that I thought might be useful that I can give you.
Definitely.
Okay.
So to get back to your degradation, if you look at the peak, we have a range of peak. We have identified tentatively those are polyethylene glycol. And as it age, it breaks down, losing one of the carbon as it goes. That's why it's like a ridge. You have peaks after peaks in a very diagonal pattern. So it helps us look for also degradation.
Mr. Avil.
So possibly in a vain attempt to stay within your 10 minutes, let me jump around a number of things. First of all, a very impressive set of wide-ranging analyses. I think, the underlying theme or a common theme that we've come back throughout the morning and the day has been this notion of how you're going to more effectively communicate this in the document. And I think that's an issue here. You provocatively identifying a couple of these things for example, in slide 13, that the spectra between the samples look different, as well as on the negative and positive sides being different. And so it raises the question of sort of how reproducible this is when you do this on the same sample, and how variable it is in the number of field samples that you have, and even raises the question of how many field samples do you have? I mean, we have some fields that are new, that are old, that have been out in the sun, et cetera, that have been refurbished. And so from the public's perspective, I think we want to know what is the range? I mean, you can analyze, you can get this incredible spectra out of one field. But what does that tell us about, what does a composite mean?
Yeah, very great comment and question. We are at the stage of identifying target, so that's why we create a composite sample to capture what are the ions or metals, what are the chemical that is in the rubber that's releasable. Once we get the target list, we'll move on to the field sample. Then we'll look at reproducibility, accuracy, the concentration versus the ID, the age across the field. Those are the issues that we have to deal with. Once we find out the chemical, we find the reference, we'll move on the field. And definitely, that's a very important question to address. But we are at the stage of what do we have before we go into the field sample. And we have repeat this analysis, this run in some of the sample, and we have persistently seeing those predominant peak. I would not say they're exactly the same every time, but most of the predominant peak, we see it most of the time.
Okay, so again, when you actually get to the communication part, most of the time needs to be sort of defined.
Okay, well, more discussion to follow the public comment period, but I think we should move into the public comment period now.